Now we will see the gene set enrichment analysis for GO categories. As we know that the library GeoStats implements some tools that can be used for the gene set enrichment analysis. For this purpose, we have to convert the microarray probe IDs into the ntrace IDs. So this is the very important step, and without this we cannot analyze the data. And we also know that these ntrace IDs, they are a very specific gene identifiers, and it is used by the NCBI for the cross-validation of data in other databases. And fortunately, the packages we are using in the R program, they are useful in mapping from probe IDs to ntrace ID. And again, we have to convert the microarray probe IDs into ntrace ID. And first, we have to convert the probe ID into ntrace ID. Then the annotation package we are using, the ntrace ID, is then converted into a new object ID, that is ALLG. And in the last step, the ALLG object is then converted into a data frame. And to obtain this target, we have to accomplish this process into a 3 steps. And the first step is the conversion the environment to a list, and the second step is the unlisting the list, and the step is the creating the data frames. So by using the 3 steps, we can analyze the data for GO categories. And we can also retain the unique entrance IDs by using these 3 steps. So let's assume that this is a HGU33A array. This is the human array by the FE matrix, and the correct annotation package is HGU133A. And the conversion proceeds as follows, because we have to use the 3 steps. First, we have to open this library, and then from this 3 steps will be accomplished. First is the ALLG, and it's converted into, we have to use the argument this HGU133A ntrace ID. Then we have to unlist this, because by using this command we are listing it. And the second step is we have to unlist this list that is obtained by this first command. To unlist, we can use this command ALLG ASDAT frame, and then we use unlist as list. And the last step is then we have to convert it into a unique row names and matrix. For this purpose, we have to store data into my IDs, and the command will be my IDs unique ALG and row names dat.s.